is this mean son of a bitch making us have a lecture on Wednesday when the very next frickin' day is a holiday? There's always a method to my madness, whether you guys realize it or not. So, take a look at our upcoming schedule. So, we are right there where it says M, have not finished diarrhea lecture. All right, we'll get to that shitty lecture in a bit. Now, if I give you Wednesday off, then that means what I talk about on Wednesday has to be moved to Monday. That means I have to give a lecture on Wednesday, the same day you have your lab final. Do you want to lecture the same day of the lab final?
No.
Would you rather have no class on this Wednesday and a lecture on lab day final?
No.
I didn't think so, right? Which is why I give you the lesser of the two evils, right? Lecture on Wednesday, no lecture on the Wednesday of your lab final. All you do is come take the lab final. That's it. Okay. You later people will be moved up a little bit, like maybe 6:30. All right, but that's why you have class on Wednesday. All right.
Thank you, John.
So that you could have no lecture on the Wednesday of your lab final. Now, if you guys really disagree with that, send me some emails, and you know, I could always just cancel class on Wednesday and create a big lecture on the Wednesday of your lab final.
Oh, yeah.
All right. So speaking of lectures and digestive system, we were talking about all the different things that take place in terms of a enterogastric reflex compared to a gastroenteric reflex compared to local reflexes due to distension of the lumen, the myenteric plexus. We talked about all these different endoendocrine cells that are monitoring the content of the chyme, releasing their appropriate hormone that goes to the appropriate target organ to do whatever it has to do. We talked about also the idea of, well, what is something that will influence the amount of stretch I get in my small intestine lumen? What would influence how fast things can move through my intestinal tract? And we tried to have this comparison of a high protein diet compared to a low protein, high plant matter diet. And I'm sure none of you really did try this. I tried. I mean, I was actually trying to save myself some time, and I ordered 2 Domino's pizzas and 4 boxes of chicken wings on Friday, so it would last me all weekend long. And by the end of Friday night, the pizzas were gone. It's like, okay, that didn't work out very well. But keep in the back of your mind the idea of when you have a high protein meal, keep your eye out for the feces in the next 24, 36 hours, and you'll find it's always a lot firmer, a lot lacking in moisture, compared to a high fiber diet, which will always be softer, larger, because of the effect that solutes have on water retention. You guys have 2 study guide questions. Pay attention to the wording of those questions. One is about what influences water coming in, and one's about retention of water in the lumen. So we know on average about 9 liters of fluid will come into the intestinal tract through various sources, saliva, bile, gastric secretions, and that will vary quite highly with what you are also consuming. If you have a liquid diet, then of course you're going to get more water than you would normally otherwise. But by the time we are getting to the sigmoid colon and the rectum, on average, we should have about 150 milliliters of moisture remaining in the feces. There could be more, there could be less, depending upon your diet perhaps. So we were looking at this thing called a villus, which is a structural feature of our intestinal tract, covered by lots of simple columnar epithelium. And they have microvilli. And we were looking at what gets absorbed across the basal side and the apical sides of these cells. Your carbs and proteins and small fats can very easily go right into the blood supply. It's the bigger lipids we're going to have problems with that we need to talk about tonight. So here they're showing you large lipid droplets not going into the blood supply, but going into your lymphatic. These little blue dots are carbs, proteins, and small lipids. They can get in there because they're small and they're not that hydrophobic. So let's slow down and do this. How do I get into a cell? We're going to be looking at mainly ideas of facilitated diffusion or something called secondary active transport. For facilitated diffusion, it is diffusion. But remember, those things can't cross the cell membrane, so they need help. That's why it's called facilitated diffusion. This specific facilitated diffusion channel is called a glucose transporter because it transports, guess what? Glucose. Down its concentration gradient. Whichever direction that is, doesn't matter. It can go either way. It simply moves glucose from regions of higher to regions of lower for the simple reason I've been hounding you guys through all 241, all through 242. When chemicals bind, bingo, you know it, right? That's a chemical, it's a protein. It has to do what all proteins do biochemically, change its shape. That releases it into the cell. Now the other one is this thing. So you got to keep in mind that when things go against the concentration gradient, in your mind, the first thought should be, oh, I need a pump of some kind. I need a carrier. Well, remember, of course, you've all have very diligently studied those sheets I put in the very first part of the course about things you should remember from 241 and Bio 160. There's two kinds of carriers. Those that require ATP and those that do not require ATP. This is a carrier that does not require ATP because it's using the effect of sodium's facilitated diffusion into the cell to stuff and force glucose against its concentration gradient. More advanced books talk about the entropy of sodium diffusion. The energy of this, right, helps me to stuff this against the concentration gradient. Now, your other issue, struggle, frustration is how the hell I keep all this stuff organized. And so, for example, my Bio 160 students this morning had their second lecture on the kingdom of Animalia and the phylums of Animalia. And I had to remind them, okay guys, first you got to go from domains to kingdoms, from kingdoms to phyla, from phyla to classes. And keep all these things, who's a class of what, or what phylum, what kingdom, and blah, 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 blah. For you, it's the same thing. What kind of a transporter is this? It's a secondary active transporter. What is the name of this? Oh, it's a sodium glucose transporter. But what kind of it is, it's asking me what is its function. And it is a secondary active transporter, which from now in your mind means, oh, it doesn't need ATP. It's a wannabe. Now let's look at, put them all together in this context. So here is a cell, and I can see there's the lumen, so there's my food. There's my 2 Domino's pizzas. There's my glucose. Wants to go into a cell, but there's more glucose in the cell, relatively speaking, than there is in the lumen, so it has to go against the concentration gradient. How can I do that? Well, sodium has more sodium outside than it does inside, so I can do facilitated diffusion of sodium through that carrier and bring sodium into the cell. And how is that possible? Because I keep pumping the damn thing out. Your normal 3 sodium, 2 potassium, sodium, potassium, ATPase pump. Always ensuring that there's less sodium in the cell relative to the lumen. And the ability of that diffusion allows me to stuff glucose into the cell. And then on the basal side is my glucose transporter, because I can then just let it fall from regions of higher to regions of lower, relatively speaking, and get that glucose in. But what allows us to run this secondary active transporter is the ATP pump. That's what allows this to work. If that stopped working, concentration of sodium would build up, therefore this carrier would not be able to work, therefore glucose wouldn't be able to come in. Yeah, I like that idea. Okay. Anyways, so this is to remind you all the various ways that things can be transported across the membrane. Do not memorize each and every one of them. Bottom line is a repeated theme. Secondary active transporter or facilitated diffusion. If you need to know more specific in your programs, you'll learn more about it when you get there. But I do want to come down to the very bottom where we didn't really get to, which was this idea of getting lipids, right, into our cell. So first remind yourself of the structure of the jejunum where most of our absorption takes place. We have plicae, we have villi, we have microvilli. I have a thing called the lacteal running right up the middle of a villus along with my capillary beds. I know carbs, proteins, and small lipids can very easily be absorbed into circulation, which then go to my hepatic portal system. But those guys have to go into the lacteal, that lymphatic vessel that is in the middle of each and every villus. So how do we do that? That's what this is going to be trying to convey, what we're looking at. The big yellow things are lipid droplets, and they're too big to be processed, and they're too big to be efficiently chemically digested. So let me ask you this. I think we've already, no, I haven't done this analogy with you guys, I don't think. I have a 3 foot by 3 foot by 3 foot cube of solid ice. 3 cubic foot solid ice. And I'm going to clock how long it takes to melt. I come back 3 or 4 days later, and it may or may not be gone by then at room temp. If I want to race between a 3 foot by 3 foot by 3 foot, I want to melt as fast as possible. I can smash the daylights out of it, increasing surface area, exposing more parts of the ice to warmer air. That, in a way, is the version of emulsification. I need to take a big, large, fat droplet and make it a lot smaller, so there's more surface area, first of all, for our enzymes. So what they're trying to show you here is the idea of our bile salts. Coming down and coating these guys and being able to make them into smaller little individual droplets. Well, look at the blue area of our diagram for a moment. What would that blue area be representing, do you think? The lumen. And what's in the lumen? What kind of material would that blue be representing, maybe?
Water.
Mucus, maybe, or the chyme, and the chyme would be mainly the water content, right? So I've got lipids in water. How soluble are lipids in water? They're not very soluble. So what does it mean about bile salts then? Are they soluble?
Because salts.
Well, all right, so that means they're soluble, but then how do they interact with the lipid? Because the lipid isn't water soluble. So your first thought should be, how is it possible for one thing to be involved in 2 different environments? One aqueous and one lipid. That comes down to the structure of a bile salt itself. So there's a term called amphipathic. And there's lots of different ways of showing it. Some people do a flat little curved molecule like this. I think it's easier just to talk about it more like this. So let's draw just a big rectangle. And then for half of our rectangle, we'll give it lines across like this. And then we'll just put some dots on this end. Just something for contrast and difference. This part is hydrophobic. Non-polar. This part is hydrophilic. It is polar. And see, I've even tried to color code it for you guys. Right? So that's what an amphipathic structure is. It has both polar and non-polar aspects to its structure. Then I go look at what a lipid droplet is. And so let's just make this a big lipid. And then look at how bile salts, before I do it, don't say it out loud, but how would bile salts orient themselves with that lipid droplet? Think about it for a second. How would they be able to do that? What are lipids found in and what are lipids themselves? What you should be thinking is, well, my bile salts could arrange themselves. We just kind of do it. We'll just do a few of them. Now the only question is, where is he going to put the blue lines and the black dots? The black dots will be on this side, because they are the hydrophobic part. They are lipid soluble. And we'll just do this as well. Just to really drum that in. And this, of course, then is water soluble. And therefore we have our blue hashes like this. In a way, it's kind of like a binding protein for lipid-soluble hormone. Now my fat droplet, and there's going to be more all over the place. It'll be surrounding the entire lipid droplet. And as the biosolids come up and attach themselves to these and start separating them out into smaller pieces, I can also trap two enzymes within this droplet. And what am I going to be able to trap inside? Well, pancreatic lipase, that came from probably the pancreas, right? And then colipase, which will help the pancreatic lipase work. And what the pancreatic lipase will do, I'll do it over here because I'm going to need more room, is that lipases in general, whether it's salivary, or the gastric we didn't talk about, or the pancreatics, they will take this structure here which we're going to call a triglyceride. Right? Tri means three. So it's these three fatty acids. And it will, and we add that with lipase. And what we will get is this idea. I'll put it right here just for the sake of room. This is a monoglycerol, monoglyceride, plus two separate fatty acids. And that's what I get when I combine a triglyceride with lipases, in this case pancreatic lipases. I will break them down into a monoglyceride and two fatty acids. Now, why are we doing that? Because this thing, well, this remind you, have you remind me remind you. This is just isn't spastic squiggling. It's shorthand. So what is, let's put it right here, what is that right there representing?
Carbon.
It's a carbon, right? And then we have, these are hydrocarbons. We're just not showing the carbons in the H2s. So remember, we're looking at what, 20 to 30 long carbon chains, maybe 60 carbons in length. Anything above 6 carbons starts to become more fat soluble, less water soluble. I have a hell of a lot of carbons here. It's way too damn big to also even fit through a cell membrane. It's too damn big. So how do I get it through a cell membrane? You do the same thing that I almost got kicked out of school for. Take it apart. Take the triglycerides, break it into small little pieces that I could then pass through the cell membrane of a cell. I'm going to reassemble the damn thing. So when we make it into small pieces, right, I need to be really efficient at pancreatic lipases, so my bile salts allow me to make large lipid droplets into small lipid droplets. The little green dots are bile salts, and trapped in there, right, are going to be my pancreatic enzymes and colipases, and that helps us to form what's called a micelle. There's these little teeny spherical droplets that are having my enzymes do their thing. So in the micelle, we're doing our lipase behavior, taking triglycerides, right, and breaking them down to make them plural into monosaccharides and fatty acids.
Monosaccharides. Monoglycerides.
Huh?
You said monosaccharides?
No, monoglycerides, excuse me, yes, not monosaccharides. I've got sugar on the brain. So in there, right, I've got a bunch of things. I've probably got some triglycerides that haven't been broken down yet. I'll have a lot of monoglycerides, and I'll have a lot of fatty acids. As they're showing you here, with that little yellow bar and that brown squiggly line, that's a monoglyceride. Those are two separate fatty acids. And as I'm running out of time to help you guys learn how to look at pictures, I realized by the interpretation of this that it looks like the green things come off of the yellow, and they get to be recycled and maybe used again, but the yellow parts might be broken down into this. That's what they're trying to tell you. Now I can have fatty acids cross the cell membrane. Now a monoglyceride can cross the cell membrane. And they will go into the smooth AR, and I'm going to reassemble them back into triglycerides. Right in there. And that's how I almost got expelled. We wanted to give our dean a car, but the car wouldn't fit in his office. So they took it apart in the student lobby, and took it in piece by piece, and then reassembled it in his office.
Oh my God.
Of course, it wouldn't fit through the door when it was reassembled.
Wow.
All right, but that's what's happening here, and that little silly joke should help you, or story should help you remember this little idea. Now, when I'm in the smooth AR, I'm going to reassemble the triglycerides. There may be cholesterol present, there may not be cholesterol present. Depends on what you had for your diet, right? But usually there is going to be cholesterol, which is what they're trying to show you here, okay? What they're trying to say is, within the smoothie AR, we're going to do this little rectangular box. We're going to combine cholesterol with our newly reformed triglycerides, and we're going to coat them in a protein. We're going to coat them in that protein. And this is my one big thing about this one author. I really like this author, but last I knew, I didn't make proteins in smooth AR. Which organelle makes proteins? The rough ER does, or free-floating ribosomes. So they should have also added maybe rough ribosomes in this. They didn't. I'm not going to worry about it for you guys, but the idea is this. Reassemble triglycerides with some cholesterols, coat them in a protein, kind of like an M&M's. I'm going to call it a chylomicron. Where does that chylomicron go? Goes to the Golgi apparatus. Awesome. Now that yellow spherical structure we have right here, that is that chylomicron. What in the heck is that blue circle around my chylomicron? That is the transport vesicle from your Golgi apparatus, right? Because I need to get it out of the cell. I need to be able to perform exocytosis. So the Golgi apparatus packages it, puts it in the vesicle. The vesicle is a phospholipid bilayer, same thing as a cell, and that's how exocytosis happens. I can now get rid of the chylomicron, get too big to go into a capillary, therefore it goes into my more porous lacteal. Remember, all lacteals join up where? Where do your lacteals all merge and hook up to? Your subclavian veins, right? Then that is your circulatory system, which then goes superior vena cava, to the heart, to the lungs, back to the heart, out through arteries, then finally to cells, maybe your liver. It's the long way. But the big idea is large lipids cannot be absorbed into circulation. They have to be broken down into small monoglycerides and fatty acids. So we use bile salts to accomplish that, along with pancreatic lipases and cholapases. I get those broken pieces into the cell. I then reassemble them back into fully formed, fully functional, large, ginormous triglycerides, package them with cholesterol, wrap them in a nice little protein coat, shove them to the Golgi apparatus so I can put a vesicle, or put them in a vesicle, and then exit them out through the basal side of the cell. Actually pretty simple. All right, this is the simple version of all of this. It can be way more complicated. We're just not even going to think about going there, because you guys don't need to go there. But any questions on the processes? All right, think about what an amphipathic structure is and the benefit of having amphipathic. That means one end can be hydrophobic, one end can be hydrophilic. One end is water soluble. It does not want to associate with lipids. The other end does want to associate with lipids, which I can break into smaller little pieces. So that's happening in our jejunum and a little bit in our ileum. We're finally going to get to your cecum, the beginning of our large intestine. And by then, we're roughly around 1,500 milliliters of moisture on average entering your large intestine. Again, it kind of varies by what you guys are eating. But as we go up the ascending, we're going to take a lot of this water out. It might take 5 or 6 hours to go up the ascending. It's a slow process. Partially because, think about one of your study guide questions you guys have for the histology part of your anatomy. It asks you to compare the mucosa of the oral cavity, the esophagus, the stomach, the small intestine, large intestine. Think about the structural design of the large intestine's mucosa compared to the genome, and that's why it takes so long. I won't give you the answer just yet. But it's going to take you 6 hours to reabsorb lots of water, maybe some bile, lots of B vitamins. Electrolytes like potassium can be reabsorbed here as well. But the big thing is B vitamins and the majority of our water is going to come out. We'll now get into the transverse colon, where we do what's called mass movements. What it really means is the bolus, the chyme, that's almost more like feces than anything else right now, gets to move a lot with each peristaltic contraction. We're talking 6, 7 centimeters maybe. It's a mass amount of movement. They're called mass movements. And they happen all by themselves about 3 times a day. One of the things that I skipped for you guys is go back to the SA node. If I were to say, how is the SA node and the DRG similar? You'd say, well, they're capable of creating their own spontaneous generations of action potentials. Well, there are actually cells in your stomach and in your intestines that have the same behavior. They automatically create waves of contraction to help move crap along. And one of those groups is in the transverse colon to create these mass movements. So I can help move this stuff along in preparation for the upcoming meals that John is scarfing down over the weekend, perhaps. We're going to go down the descending colon, and by now we're probably down to a couple 100 milliliters of fluid. We're going to get into the sigmoid, and we're going to find ourselves in the sigmoid colon and possibly the rectum. So there's our rectum. There's our feces. And you first get your bearings and go, okay, yep, there's internal sphincter, external sphincter. I remember the type of muscle tissue, which is what for the internals? And the externals are? Awesome. Now, nobody say anything out loud. Nobody out loud. But I've got baroreceptors, which are stretch receptors, and I'm familiar with them for the heart and for breathing and stuff. And they're going to get stretched, and they're going to send a signal, which means these have to be sensory neurons. My question to you is, does anybody remember the structural classification of that neuron? We hate to be ashamed to waste all that knowledge you guys have crammed into your head. How many of you said, oh, dude, that's easy, that's a unipolar neuron? Yes, because it's a unipolar neuron. Remember, unipolar neurons are sensory neurons. I know sensory neurons connect to what part of the spinal cord? Oh, dude, that's easy, the posterior gray horn. Because the posterior is sensory and the anterior was motor. And the motor has 2 kinds of motor neurons, the visceral and somatic. Oh my God, all these nightmares just flooding back into your heads, right? So we're supposed to be remembering all the time, right? So, stretch, action potential, up my unipolar sensory neuron, goes to the posterior gray horn and synapses with a visceral somatic motor, right? From the anterior gray horn, and they even put it in the proper place because the visceral is near the lateral gray here. The action potential then travels down, goes to my internal sphincters that are smooth muscle tissue, causing them to relax instantaneously. Now take a look at that one, take a look at this from here. All right, so follow my red light. What kind of a reflex pattern is that? Very good. Monosynaptic. There's only one synapse in the whole pathway. And what's the cool thing about reflexes? They don't have to send messages to the brain. It's a bypass. The fact that you are aware of it is a total different sensory path. But what I want to add to this is there's the reflex arc to relax the internal sphincters. And what they should show you is another sense up here going all the way up to your somatic motor cortex in the postcentral gyrus, making me aware that my rear end wants me to poop. And then I go, well, I'm lecturing right now. I don't think my students would appreciate that, right? But that's your conscious control. That's what they mean by this, right? Somatic motor, right? This is conscious control. The cerebral cortex is where you give permission to the externals to relax. So just remember, there has to be probably another sensory route to the cerebral cortex, so you're aware of the fact that you need to poop. And then that gives you the conscious control of being able to relax those external sphincters. All right, let's talk about more poop. Feces and their composition and what the different types are. So how many of you work in nursing stations where you've had to work with individuals who are bedridden, maybe wear diapers, have that bedpan, and you have probably, for the most part, seen the entire array of different types of fecal material. I once had a lady named Harriet. I will never forget Harriet. I was a high school kid, and this is way before you had to be trained as a CNA. You just show up first day, I want to be a CNA. Great, here's your white uniform. Good to go, let's go. And I was working graveyard, and I switched to mornings and went into the morning meeting. The charge nurse said, Harriet has a present for you. She drew you a picture. Oh, that's nice, that's great. Oh God, it was not great. Harriet only had one kind of medium to paint with. That was her feces. And she painted on the entire wall and her face. And it was me who had to clean it all up. All right? But I very much remember now what type 7 looks like. Type 7 is the rating that we give to feces that is a pure water content. And you can even get a Bristol Stool t-shirt that has all the rating scales of feces on it. Nicole wants to buy one already, I can tell. Right? And so we organize it by the idea of sort of this. If we zoom in a bit more here, I think I did this with, here we go. All right? If it almost has very little water content at all, it could be a type 1, like rabbit pellets, right? All the way into your normal little healthy ones, which should be really, yeah, between 4 to 6 is on average, somewhere in there with that. But how do we really get to this really severe watery type of diarrhea? Well, there's two kinds of, or two categories of diarrhea. There's osmotic and there's secretory. And they're both, in a way, really the same. It's the cause that's the difference. Go back to someone who is lactose intolerant, right? Meaning they cannot absorb the sugar lactose. Their genes no longer code for, well, they code, they just don't make the lactase enzyme. So it means lactose is in their lumen, it's a solid that I cannot absorb, it acts as a water attractant. And the more lactose I have, the more solid particles I have, the more water I will attract into my lumen and keep in my lumen. The more water I have, the more volume I have. The more volume I have, the more stretch. And the more stretch, the more local stimuluses I get for more peristalsis, the faster it moves. And the faster it moves, the less time I have to absorb the nutrients, kind of like Lucille Ball chocolate factory idea, which is why you get diarrhea. Because it simply, A, can't be absorbed, and if you could, it's simply just moving through too doggone fast. That is osmotic diarrhea. It is diarrhea due to the osmotic draw of water due to the foods that you eat and the things you cannot absorb. Now, secretory, it's the same idea. It's due to solid particles in the lumen. But it's the cells that secrete the particles. They're secreting ions, mainly chloride ions, into the lumen to make it hypertonic, which would attract water, volume, distension, faster movement, diarrhea. Because your body's saying, get this stuff the hell out of me. So if I secrete ions into the lumen, I will make it more hypertonic. I will cause more water to be drawn in, and therefore I will get diarrhea because of it. So keep in mind the difference was osmotic is more like a dietary issue, things you've eaten or can no longer absorb normally. But secretory is more of a pathogenic response. Like if you have giardia, because you drank out of a stream. Now let's look at this. Look at some alternative ways of trying to lose weight. Lap bands are pretty common, but it's still severe surgery, anesthesia, and risk of patient dying during the procedure. But the idea is this lap band will go around the cardia portion of the stomach and make that now my stomach. And you'll have a little tube that goes to an inflatable pouch, to a port that's outside your abdomen. You can literally put a syringe into it and add more fluid to make this tighter. You can literally draw some fluid out to make this a bit looser to increase the volume size. And by increased volume size, I mean maybe adding an extra 2 tablespoons. Where now normally your diet might be at 3 tablespoons, you feel full. And you can rupture it, you can overeat, you can screw it up, but it can also work very nicely for some people. I had a student 2 years ago. She lost over 300 pounds with one of these. She was very upfront about it. And she said, when I had it removed, it was all up to me now. Now look at me now, remember how I was then? It's like, do I want to go through this all over again? And she said, hell no. Hopefully she's maintaining her new lifestyle because her weight gain was simply just due to overeating. Now that makes complete sense, right? You're just making the cardia portion a small little stomach, but everything is normal from that point on. Now don't say anything out loud. Let's test your critical thinking here. Look at the gastric bypass. Study it for about, say, 30 seconds or so. What is and what is not happening here? Why does this work? Think about it physiologically. You guys know already. All right, they're going to come in, and we're going to keep all of this. Nothing's going to be taken out. If I look at this, I realize continues to the small intestine, and there's this thing. So I'm kind of thinking that whatever this part where my red light is, they cut it off, and this part is now that part. They just cut it, and then it went all the way up here. And then it looks like they kind of cut the cardia portion out of my stomach and left the fundus in the body and the pylorus and the duodenum and the jejunum, a good part of it. Left it there. So that apparently is my new stomach. So what is my chyme not going to experience? What have they not allowed access to, that chyme?
Everything.
To absorption.
So what do you mean everything, Scott?
Yeah.
Am I going to have very many chief cells in my new stomach?
No.
Am I going to have very many parietal cells? Am I going to have any G cells for gastrin production? Am I going to have any D cells for somatostatin? No. I mean, you've lost all this hormonal regulation. And then what I think I heard David say is, well, is that chyme then going to go right to my duodenum?
Well, no, I was saying you lose the duodenum because they cut it off, right? So you don't also have that
No.
signaling.
So, right, hang on. Right, you're right. All right. Because Scott was going, no, but Scott, there's your duodenum. That's not your duodenum. You completely bypass your duodenum. Okay, big deal. Well, what goes into the duodenum? Well, something called the hepatopancreatic duct, which means bile, pancreatic juices, and it ain't going there either. And what's the cool thing about the jejunum? A tremendous amount of surface area, plicae, villi, microvilli. I'm probably bypassing all of that as well, which means I'm not going to get much nutrients out of whatever it is I am eating, which is why this works so well. You bypass the hormonal and chemical regulation. You bypass most of the surface area. So you have to be probably really diligent in what you eat, right? How much of it you eat. Then realizing you're missing most of the factory production of being able to get the nutrients out.
So they're cutting it below the jejunum?
Probably the latter part of the jejunum.
Wow.
How long is the jejunum? Do you remember?
about 8 to 10
It's
feet.
about 8 feet, yeah, so maybe leave it. And this is where surgeon choice comes in based upon what we need to do with you. You know, give you the last 10 inches, give you the last 1.5 feet, give you the last 2 feet. Depends on how much of a naughty boy you are, kind of a thing. All right? Now, my sneezing's catching over here.
Would it be easier to get sick that way?
Huh?
Would it be easier to get sick that way from food?
Maybe, maybe not. I don't know. Probably since we're now realizing how much intestinal bacteria play a role in our health, it might not be the best idea to do. Then compare that now to this idea over here. What do they do? How is this different from that? Well, they basically kept my stomach. They just only gave me half of a stomach. I have all the parts, just not all the parts. I still have a duodenum, jejunum, so I still have my connection to the gallbladder or the pancreas. And they might even have options of rerouting your pancreatic duct and your bile duct if they think you're going to need it. But pretty much, remember, that goes in right there. So this is just giving you a smaller stomach, so you eat less, right? But all the connections, see, there's the bile duct right there, are still intact. Now, what's interesting is sort of how a lot of this knowledge came about. So once final exams are over, and you want to relax, curl up with a nice little good book to read on a Saturday. Read Mary Roach's Gulp. It's a Saturday read. It's a quick read. It's about, as she calls it, the journey through the alimentary canal from mouth to anus. It's about the history of our understanding of the digestive system. The history of fecal transplants. Back in the 70s, you literally had a bucket of poop in the back of the truck, kept on ice, and then had to be purified in a micro lab and then sent back to the recipient and kind of stuff. You get to learn how Elvis Presley actually truly died and why he actually died. But also this idea up here. One of our first understandings of how the stomach works was back in the 1700s when a soldier got shot with a musket ball that just tore a hole right into his side. And they couldn't close it. There was not enough tissue to close it. And surgery was still relatively primitive back in those days. Well, Dr. Beaumont thought, oh, that's a shame. That's kind of cool. I wonder how the stomach works. Let's use you to figure that out. I'm going to tie a piece of pork on the end of a string and then just poke it into your stomach, and every hour take it out and see what happened to it. I'm going to have you swallow something, and I'm going to be looking at it as it comes down your esophagus and how it hits the stomach. I'm going to just make that a little window into your stomach. Well, that should sound maybe familiar to some of you if you work on a farm. We do it to cows all the time. Give them a fistula, a permanent opening to their stomach, so I can literally reach in, grab their chyme, and see how well they are digesting the alfalfa, the oats, the grain that I gave them. It's a permanent opening. They don't get any infections, right? They're on antibiotics, and they're very well monitored and cared for. But if you ever see a cow walking around with this big plastic little toilet-looking like thing, that's a fistula. And it's there to see what's happening to the contents of their food, to see if we got too much of whatever that they're trying to scientifically balance with cows. Now, this is not in your notes upcoming, but just to give you an idea of, again, how much the stomach can hold. And I might just send you guys a clip to this just because I find it really interesting. I'm intrigued by some competitive eaters, and there's a guy whose video I watched on Saturday to see if it's appropriate for you guys to look at. And this guy will do any challenge you send to him. And he ate a daily diet of some world heavyweight power lifter. But he ate it in one sitting. He ate it in an hour. What he ate was 1, 2, 3 family-sized Papa John pizzas, 2 trays of brownies, and a large cookie, all in 50 minutes. Plus 2 liters of Dr. Pepper or something. Okay, I mean, oh my freaking God. All right, but again, what is your stomach for? It's really not for digesting. It's a storage bag, right, to hold the food. And another national context, the Super Bowl of eating championships is Nathan's hot dog eating contest, where the current winner ate 72 hot dogs and buns in 10 minutes. 72 hot dogs and buns in 10 minutes. Right? And what do you consume? About 8.6 pounds of food overall, right? 18,000 calories, right? 1,200 grams of fat, 48,000 grams of sodium. I mean, it's just, and these guys go eat dinner later that night. And they have breakfast the next morning. I mean, remember, these guys train sport, that they train, and they're actually very, 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 for the most part, fit. And when you watch these things, I've been noticing this, the skinnier they are, the better they do. Because there's less abdominal fat to impede stomach expansion, which is why these really skinny guys can eat so much. Now, how many of you, to help you move things along a little bit, or maybe have a relative or someone, and you have them have some prunes, right, to help keep regular? Why do prunes make you regular? Because they contain sorbitol. You cannot absorb sorbitol. Therefore, it acts as a solvent to attract water. But don't eat more than 50 prunes in one sitting. It's kind of hard to do, but that's going to give you way too much sorbitol. And you'll get diarrhea if you overeat with prunes. How many of you like Bing cherries? They contain sorbitol, which you might remember, oh, that's why as a kid I always get, because I always ate way too, 2 or 3 bags of cherries at once. So it means you got way too much sorbitol.
That's why they say you're not getting so much cherries.
How many like pears? Pears have sorbitol. So maybe you can have a little bit of prune for breakfast and some cherries with lunch and maybe a pear with dinner. You'll get a nice amount of sorbitol to help keep you regular to a certain extent. And then there's apricots, also contain sorbitol. So you got prunes, you got cherries, you got pears, you have apricots, all containing sorbitol, a chemical substance that occurs naturally that we cannot absorb. So the more you consume, the more moisture you can have in your feces, and you can kind of self-regulate a little bit, maybe for your own personal physiology. Now, interestingly enough, about prunes, the California Prune Association used to say that there's a myth that dried prunes, cause diarrhea. And they're saying, no, it does not cause diarrhea. And then this is the California Dried Plum Association. When you click on their website, all you get now is a 404 arrow sign. They were told to take off their statement because it is not a myth. It can actually harm somebody. That if you get too much diarrhea, you'll be dehydrated and cause all kinds of problems. But maybe you don't like prunes or cherries or pears or apricots. Maybe you like something a little more sweet, a little more out of a vending machine. Gummy bears. Do gummy bears cause diarrhea?
Yes,
Oh, yeah.
they have sorbitol.
No, not. You're close. You're close. They have what's called lycasin, which comes from mannitol, which is, again, an unabsorbable sugar. So lycasin is the hydrogenated glucose syrup. If you see that, it means lycasin. If you see lycasin, it means you cannot absorb it. So after about 50 gummy bears, you're going to be heading to the toilet.
There's a guy on YouTube that eats that bag of gummies. He's on the toilet for a while.
Yeah, okay. Smart man, okay. All right, but that's why it works. Shoot, we're all done. I should add flatness to this lecture.